Hello, everyone. Welcome to the Interagency Modeling and Analysis Group Multiscale Modeling Consortium New Investigator webinar. I am very pleased to introduce the speakers for today. The speakers are Dr. Jaron from Drexel University. Dr. Jaron is Calhoun Distinguished Professor of Engineering in Medicine at the School of Bioengineering and Health Science and Health Systems. It's interesting to note that Dr. Jaron led the engineering team that developed the intra-aortic balloon pump, the first clinically successful cardiac assist device, and produced models of cardiovascular systems dynamics to understand the device's interaction with the circulation. More recently, Dr. Jaron's research has focused on elucidating specific complex mechanisms of the microcirculation by creating models that integrate biochemical and mechanical interactions in the system at differential hierarchical levels. Currently, his work is focused on mechanisms of production and transport of nitric oxide in the microcirculation. And on your screen, to the left of Dr. Jaron is Dr. Kenneth Barbee. And Dr. Barbee is Professor and Deputy Director of the School of Biomedical Engineering Science and Health Systems at Drexel University. Dr. Barbee was among the first investigators to use atomic microscopy to image living cells. His studies are focused on cardiovascular mechanotransduction, focused on the effects of acute enrichment of endothelial cell membranes, signaling events related to nitric oxide production. And finally, to the left of Dr. Barbee is Dr. Donald Buerk. And Dr. Buerk is research professor at Drexel University and is a senior scientist at Medvas Concepts LLC, where he is helping to develop targeted nanoparticle systems for delivering anti-cancer drugs and other cardiovascular related projects. So we are so pleased to have you present the U01 New Investigator webinar. This particular U01 award was funded by the National Institute of Heart, Lung, and Blood. And the presentation is entitled Multiscale Transport Dependent Nitric Oxide Signaling Cells to Vascular Networks. You're welcome to take it away.
Thank you very much, Grace. It's a pleasure to make this presentation. And you already know what the title of the presentation is. Let me just explain why we have three people making the presentation. The reason is that this is an integrated approach to the research where we have modeling, we have in vitro studies, and we have also in vivo studies. All of them complement each other in the research project. So in the presentation, I'll be talking about the work which led to the current project. That's work that was supported both by NSF and by NIH at different stages. I will discuss some of the early research by our group, and I will talk briefly about the specific aims of the current project. Dr. Barbee will take over, discuss some of the hypotheses that we are posing. He will talk about some of the innovative approaches that we have proposed. He will discuss the real-time in vitro measurements and discuss the progress on the cell scale model that we are developing. Dr. Donald Buerk, then, who is responsible for the in vivo measurements, will discuss his approach to the in vivo measurements and some preliminary results relating diameter of the vessel to nitric oxide concentration. So what we have done is, over the years, is we have used stepwise methodology in order to approach the problem. We started by looking at nitric oxide transport through multiple vessel layers in a single vessel. We then coupled nitric oxide transport to oxygen transport. We examined then the effects of hematocrit and the two-phase nature of blood on both nitric oxide and oxygen transport. Later on, we coupled nitric oxide and oxygen transport to the effects of blood flow, included convective transport, which turns out to be a very important element in trying to understand the transport of nitric oxide. We then went on to study a pair of arterial and venular to see the effects of the venular system on what happens in the arterioles. We then added capillaries to the simulation, and finally we extended the vascular network, and we extended it to the vascular network scale. We started developing a cell scale model now, and Dr. Barbee will discuss that in more detail later on. So the first step, we started to simulate the interactions between nitric oxide and oxygen in arterioles. We looked at what happens in the blood vessel lumen, in the plasma layer, in the endothelial layer, and in the vascular wall, and finally in the surrounding tissue. To do that project, to do this simulation, we used finite element computational modeling, and we began by looking only at a steady state. The dependence between nitric oxide and oxygen we simulated using Michaelis-Menten kinetics. The nonlinear diffusion reaction equation was used in order to model the system, where we included production, convection, scavenging, diffusion, and consumption. So here is the a diagram of the vessel, and on the right you can see the various layers that we studied, all the way from inside the vessel to the periphery of the tissue. So we have, at the beginning, we have the RBC-rich core, the poor plasma, etc., all the way to the tissue. As I said before, the governing equations to study this particular system were the nonlinear diffusion reaction equations. The NO production dependence on O2 was simulated using Michaelis-Menten kinetics. In the vessel lumen, we looked at the hemoglobin as the primary scavenger for nitric oxide, and the oxygen availability in the vessel and in the tissue was determined as a function of hematocrit and hemoglobin. In the endothelium, the synthesis of nitric oxide from eNOS was simulated as being oxygen dependent, and again using Michaelis-Menten kinetics. So the production rate of nitric oxide was calculated using the endothelial oxygen concentration profile. In the vascular wall and the tissue, nitric oxide was primarily scavenged by soluble granulate cyclase, and nitric oxide turns out to have an inhibitory effect on oxygen consumption. This is an example showing nitric oxide profiles using oxygen-dependent nitric oxide production. So what you have here on the left, on the y-axis, is the concentration of nitric oxide. On the x-axis, you see the distance from the center of the vessel all the way out to the tissue. The scavenging rate inside the vessel was fixed at 382.5 per second. And as you can see, as we vary the scavenging rate in the tissue with very low scavenging rate, the nitric oxide concentration is very high, but as the scavenging rate increases all the way up to 10 per second, the NO concentration decreases considerably. So the scavenging rate in the tissue has a profound influence on nitric oxide concentration profile. The next step that we used in the process was to include the Farreos effect. And the Farreos effect has a cell-free layer next to the vessel wall. We varied the thickness of the Farreos layer from 1 to 6 microns. And of course, in that section, there is no scavenging by hemoglobin because there are no RBCs. Factors which we included, which we considered in this particular element of the model, was we looked at different blood velocity profiles. We varied the thickness of the plasma layer. We included the presence and thickness of the endothelial surface layer, the glycocalyx. And data for this simulation was taken from experiments reported in the literature. Here is again an example of oxygen, mean oxygen concentration, mean oxygen PO2, and peak nitric oxide as a function of the thickness of the plasma layer. So if the solid line depicts the plasma layer when you have red blood cells in the plasma layer, the dotted line represents the effect when you don't have any RBC in the plasma layer. Now look, this is the nitric oxide, this is the oxygen, and you can see that if you don't have red blood cells in the plasma layer, you can see that NO increases significantly. And of course, if you do have RBCs in the plasma layer, NO concentration is decreased considerably. And the opposite effect is observed on the oxygen. The results that we got also suggested that oxygen availability to distant tissue increased because greater inhibition of oxygen consumption by NO. This is not shown here, but this is one of the observations that we made. We also observed that the Farreos effect becomes more significant in enhancing nitric oxide bioavailability in vessels with diameter less than 30 microns. The next step was to include a few red blood cells in the plasma layer, and when that happened, the NO level was decreased significantly, and the PO2 level increased only slightly. Also, blunter blood velocity profiles turned out to be associated with significant increase in oxygen delivery to tissue. The next step was to study paired arterial and venial. Here is an image of a pair showing the arterial and the venial and a very rich capillary network. This is taken from Kobayashi and Takizawa. And the features which we included in this step in the model was that NO is produced both by the arterial and the venial, as well as released by the capillaries, and NO diffuses from arterial to venial. The capillaries in this particular model were modeled as a porous medium. Here is an example showing oxygen distribution. On the right is without capillaries. On the left is oxygen distribution with the capillaries. And you can see that the capillaries do contribute to oxygen distribution, but not very significantly. In contrast, if you look at the NO distribution without capillaries on the right and with capillaries on the left, you can see that the NO distribution is enhanced considerably when you model the capillaries in the system. So the effect of capillaries is greater on nitric oxide than on oxygen. And as a matter of fact, it turns out that in the presence of capillaries, nitric oxide concentration can be higher in the venial than in the arterial wall, especially when the vessels are very close together. This has been confirmed in part by our own in vivo measurements already. The next step was to move to a vascular network and its relationship to shear stress. This is the geometry of the model. This is the hamster cheek pouch retractor muscle. You have a three-dimensional representation, and you can see two arterioles with branches in the system and a system of capillaries. This is an example. We are showing here two arterioles which are branching at a certain point, and we are plotting nitric oxide concentration. Now, it's easier to see the concentration in the bottom figures right here. This is measured, or this is computed at this level right here. The middle one is computed at the middle level, and the one on the right is computed at the lower level. Now, it turns out that NO distribution in the vascular bed is related to both NO production due to shear stress and the RBC phase separation effect. You can have a, just lost one of the, I'm turning it back on. You can have a vessel branch which has a greater fraction of blood flow from the parent vessel. That also always tends to produce larger shear stress on the vessel wall. However, if there is a larger number of red blood cells that enter this branch, they cause greater scavenging of NO in this branch, so that you can have actually a decrease in NO concentration in that branch. So the goal of the new this is all past history. The goal of the new project is to develop a multi-scale dynamic mathematical model of NO production and transport. We need to refine and integrate cellular, vascular, and tissue scales and validate predictive features using experimental data. And the methodology that we have proposed to use is to integrate signal transduction and flow-dependent production and transport of nitric oxide and include the effects of oxygen, hemoglobin, calcium, ATP, nitrite, and other chemical species that determine NO bioavailability in blood and tissue. We have to do that by linking lower to higher scale models, validating at each step by applying innovative techniques, which are going to be described in a few minutes by Dr. Barbee. And we have already started performing experiments to support the development of the cell level model. The specific aims for the project at the cell level determine the role of ATP autocrine stimulation and calcium dynamics on flow-dependent nitric oxide production. At the vessel level, determine the role of nitric oxide transport in the dynamic regulation of vascular tone and blood flow. And at the tissue level, determine how oxygen deficiency modifies NO production and in turn regulates vascular tone, blood flow, and oxygen delivery to tissue. In order to do the cell experiments, we developed a very unique chamber, which is going to be discussed again by Dr. Barbee. And in order to make sense of the measurements of that we make in this chamber, we had to produce a special model that explains the measurements that we make to the actual NO concentration. These measurements are going to be very critical in order to develop the cell scale model, and the in vivo measurements are going to be used to validate our model and to link the different scales. The scales, the way we propose to link the scales is the cell scale to vessel scale through flow-dependent mass transport of oxygen, ATP, and nitric oxide in the bloodstream, and then the cell to vessel and to network scales using the dynamic relationships at the cell vessel level between shear stress, oxygen, nitric oxide, nitrite, and ATP, and their effect on the pathway between cGMP and smooth muscle tone. I will now let Dr. Barbee take over, and he will continue with the presentation.
Thank you. So our in vitro experiments are done with a parallel plate flow chamber, and I'm showing this schematically here to illustrate the transport phenomena that we have to consider to understand our measurements and also to inform the design of our chamber. So if you have flow through a channel with cells on one side producing something, whether it's nitric oxide, ATP, or anything else, then there's a concentration profile that will develop along that wall, increasing in the direction of flow. If then suddenly the flow rate is increased, then the concentration at the wall will go down if the production is a constant. So that the convective washout of the produced and released substance causes a decrease in concentration. On the other hand, though, if the increase in flow and the attendant shear stress increase stimulates an increase in production, well, then it's not clear whether the concentration of the wall is going to go up or down. And so we model that. We have a diffusive convective transport model for the flow chamber that makes the following kinds of predictions. So the curve in the middle, the thick black curve is the concentration profile under a very, very low flow rate. And you can see that the concentration of NO extends pretty far into the lumen of the channel. With only small increases in flow, however, all of a sudden the concentration at the wall goes down and the concentration gradient near the wall becomes very steep. Further increases in flow and the stimulated increase in production eventually result in an increase in the concentration at the wall. And a summary of that is shown in the inset, where the top curve is the concentration of NO at the wall as a function of shear stress under steady state conditions. So you see it goes through a minimum for very small changes in flow. There's always a decrease in concentration, and only when the flow stimulated production increases sufficiently to overcome that convective washout do we get an increase in concentration at the wall. So this has informed our design of a chamber to make measurements of NO. In particular, the steep gradient at the wall suggests that trying to place an electrode near the endothelium, hovering over some distance from the cell surface, is problematic because of the steep gradient there and the near impossibility of accurate positioning there. Not to mention that the electrodes tend to be flow sensitive, and they themselves would disturb the flow. So our solution to that is to design a chamber in which endothelial cells are grown on a permeable membrane, and the electrode is placed on the abluminal side of the membrane. So the electrode is placed a fixed distance from the endothelium and is isolated from the flow in the chamber. So this allows us to make direct real-time measurements of NO concentration at the cell layer. And shown on the left here is a family of curves showing the response to different size step changes in shear stress with a stress-dependent steady state level of NO. Notice also that the kinetics of the response are different depending on the level of shear stress, with a faster rise time for the higher shear stresses. Note also that at the time point that the flow is started, that's at 50 seconds on this chart on the left, there's always a decrease in the concentration at the wall as predicted by our model for convective washout. And so this suggests that the kinetics of the ramping up of NO production is time dependent, as indicated by these curves. Now we can use a model similar to the one that I just showed you in order to back out NO production rates. So these measurements of NO I'm showing you are only deltas. They're changes from the steady state, and we don't have an absolute measure. However, we can model the response in order to back that out. And to show that, it's a little bit complicated figure here on the left. The NO concentration at the wall is proportional to the production rate, only the slope of that relationship depends on the flow rate or the shear stress. And so by plotting, knowing the delta between each of the steady state shear stresses, we can place those data points on these curves and back out a relationship. So by fitting the data for a range of shear stress steps, we can determine the best fit functional relationship between production of NO and the shear stress shown in the lower right corner. And as indicated there, there's a hyperbolic expression that does the best job of fitting RNO to shear stress. And doing this kind of a fit allows us to unambiguously determine the basal production rate that we're starting from. So now to get to why we designed this whole thing is to study the endothelial cell response to flow. And our previous work suggested that an important component of the shear stress response was actually mediated by agonist stimulation by ATP that is actually produced by the endothelium. So it's been known for some time that shear stress induces ATP release. The release of ATP is calcium dependent. Calcium ATP stimulates calcium release, and there's a positive feedback that leads to further ATP release. And so the concentration of ATP that's achieved at the cell surface depends on the release rate, the extracellular flow conditions determining the convective transport, as well as the binding reactions at the cell surface and the degradation of ATP. Intracellularly, we find that there are also some transport issues that are involved in the signaling process, and we're particularly interested in what goes on at membrane structure called caveoli, which are small invaginations of the size order of 50 to 100 nanometers. These are the locations where eNOS is located, the enzyme that makes nitric oxide. Caveolin 1, which is the protein necessary to create that invaginated membrane structure, and also binds with eNOS in an inhibitory interaction. Calcium binding to calmodulin interacts with eNOS to cause release from Cav1 and the beginning of production of nitric oxide. eNOS can also be activated by phosphorylation steps, which we'll talk about in just a minute. But the calcium dynamics are the interesting part to us, because the binding of ATP to cell surface receptors triggers a release of calcium from the endoplasmic reticulum. And simultaneously, there's an activation of plasma membrane calcium channels, so-called store-operated channels that are triggered by the loss of calcium from the endoplasmic reticulum. And there are a couple of mechanisms that have been proposed for that, and we'll talk about those in a minute. The way we study this, the difference between calcium that's released from the ER versus calcium influx through these membrane channels that happen to also be co-localized to caveoli, is doing an experiment in which we stimulate with ATP in calcium-free medium. So the ATP stimulates release of calcium from the endoplasmic reticulum, but there's no influx from outside because we removed extracellular calcium. The depletion of the ER, however, will stimulate the plasma membrane store-operated channels, so that when we then come back and add calcium, we get a big influx through these store-operated channels and a sustained elevation of calcium. So the dynamics of the calcium response shown on the top figure. In the bottom figure is nitric oxide fluorescence, and this is not using our electrode. This is using a fluorescent dye called DAF2 that doesn't give us real-time measurements of concentrations. Rather, it reports the accumulated release of nitric oxide. So the slope of that line is indicative of the release rate. And doing a simple relationship between the calcium load and the NO produced, we can see that the production of NO due to calcium influx through the plasma membrane channels co-localized in the caveoli with the eNOS is much more efficient at stimulating NO production. The blue curves are showing the same experiment when a calcium chelator has been introduced into the cytoplasm, and we can see that it has an effect on the calcium and the NO production, but again, looking at the relationship between calcium load and production of eNOS, we still see that the influx through the extracellular channels is more efficient at stimulating NO production. And there's a little animation here showing how that works with BAPTA. In this case, we're modeling an experiment with baptagargan causes a release of calcium from the ER. The calcium then has to diffuse some distance to get to where the eNOS is. If there's BAPTA in the cytoplasm, it's going to severely restrict the amount of calcium that reaches the eNOS. But at the same time, now calcium is going to enter through the store-operated channels where it can still activate eNOS. And we've modeled this situation with a mathematical model that incorporates all the calcium signaling dynamics. I'm not going to go into detail on all these things. We have diffusion of calcium in the cytoplasm, buffers for calcium, calcium release mechanisms through the IP3 pathway, which has been modeled by others, various pumps that remove calcium from the cytoplasm, and finally the calcium entry through these store-operated channels that are co-localized to these caveolar domains. Now, the geometry that we use is rather idealized, as we tend to always do, and so we're modeling the caveolar pathway, the influx through caveoli, as a small domain in the whole cell membrane. So we have ER release from a central compartment and extracellular influx, not from the whole plasma membrane, but just from a confined region. And just noting that that confined caveolar domain is not a single, not meant to represent a single caveola, but rather a cluster of them, or just a domain in the membrane with caveola. So, so here's a simulation of the calcium response when calcium is released from the ER, and this color scale on the bottom makes it look like there's a big gradient, but the actual difference in the concentration from the ER surface and the plasma membrane surface is actually very, very small. If you probably maybe can't read those numbers, but it's tiny. The graph on the top shows the overall calcium with the experimental data, and we adjusted the fluxes in order to match our experimental data. The graph on the right shows the calcium concentration at different locations in the cell, and it looks like one line because all the curves overlap. And so there's really very small calcium gradient when it's released from the ER and can diffuse everywhere. When we model influx through the confined area where the calcium extracellular CCE channels are, we then start to see these sharp gradients in calcium, and indeed the concentration in the vicinity of the caveoli where eNOS is significantly higher than the bulk average calcium, which, by the way, is what people typically measure when we measure cytosolic calcium. The graph on the right is showing the concentration curves at different distances away from the plasma membrane. So the top one is right in the neighborhood of the caveoli. We see that this is showing what happens if we take that domain where the caveoli are and spread it out over the whole membrane, keeping the flux constant. So just spread, instead of being confined to one small area, we spread it out over bigger areas. And what happens is that we lose that concentration effect. The concentration at the level of the membrane looks like the concentration in the bulk cytosol. Another consequence of this geometrically determined calcium profile and signaling efficiency is that the shape of the cell could also potentially affect the calcium distribution and potentially the efficiency of the signal transduction. And in particular, we know that endothelial cells align and elongate in direction of flow. There's also evidence that the caveoli tend to cluster at the ends of the cell, particularly the upstream side. In this case, we modeled at both poles of the cell. And by looking at the calcium concentration as a function of the aspect ratio of the cell, we see that the effect of segregated caveolar domains is enhanced by this change in geometry of the cell. Okay, so that's our model of calcium signaling inside the cell. What about nitric oxide production? Well, when we look at the NO response in our flow chamber, these are some of our data when we interfere with those two important mechanisms that I discussed. So one is the extracellular autocrine stimulation by ATP that's released by the cells. And in one example here, the top graph, we're showing what happens when we block the purinergic receptors on the cell surface. So we interfere with that feedback signaling of ATP acting on the cell surface, and we indeed see an inhibition of the NO response to shear stress. In the bottom figure, we show the response when we block the store-operated channels. So SKF is a channel inhibitor that blocks store-operated channels. And again, we see a fairly significant decrease in nitric oxide produced in response to changes in shear stress. So both of those mechanisms seem to be important for the NO response to flow. Going back to that experiment I spoke about before, where we stimulate with ATP in the absence of extracellular calcium, and then we add back the calcium. So this is to show explicitly the dependence of the CCE, or store-operated channel influx. So this top graph, the top curve with the solid triangles, shows the eNOS phosphorylation response to ATP stimulation. with extracellular calcium. If we stimulate with ATP without extracellular calcium, we see that that phosphorylation is reduced. It's transient and it recovers back to baseline. But then we go back and we add back extracellular calcium. So we would now get an influx of calcium through those store-operated channels. We again see a sustained, significant increase in the phosphorylation of phenols. So now a similar experiment where now we're stimulating with ATP in the presence of extracellular calcium, except now we're blocking those store-operated channels. And again, we see that store-operated channel calcium influx seems to be important for the eNOS phosphorylation in response to at least agonist stimulation. And on the right, you see that the blocking those channels does indeed inhibit the calcium influx, particularly in that sustained steady state regime. So now we wanted to look into some of the mechanisms governing this store-operated channel pathway. And one signaling molecule that's been implicated is protein kinase C, which has been suggested to act on play a role in the stimulation of the store-operated channels. It's also calcium dependent. It depends on interaction with calcium and diacylglycerol, which are part of the front end of that signal where IP3 is generated in response to agonist stimulation. So one way of investigating this is to just inhibit protein kinase C with an inhibitor called chelarethin. And when we do that, we see that we significantly inhibit the calcium influx. So it does seem to play a role in that capacitive calcium entry. And when we inhibit PKC, we also see an inhibition of the phosphorylation of eNOS in response to ATP stimulation. And I'll come back to this PKC mechanism in a bit. The last thing I want to touch on in this talk is to now turn our attention to a pathological condition, and that is hypercholesterolemia. And when we think about vascular disease and cholesterol, most people's mind jump to the formation of plaques, so the accumulation of LDL in the intima and infiltration of macrophages and development of lesions, which can eventually occlude the vessel. And this is the famous graphic from Michael DeBakey about the prevalence of these kinds of lesions in different parts of the vasculature. I want to focus on a different piece of the cholesterol story, and that is we know that cholesterol is a component of cell membranes, and this is data from a colleague, Tom Palenko, looking at the enrichment of plasma membrane cholesterol in an animal model, that a fat-fed rabbit model. And you can see that over the course of days, the serum cholesterol and the membrane cholesterol track each other pretty much one-to-one. We also know that there are daily fluctuations of blood cholesterol levels. The postprandial period, there's an increase in blood cholesterol, and during the same time window, there's an impairment of flow-dependent vasodilation. So we wanted to look at the effect of cholesterol enrichment of the membrane on the shear stress response of endothelial cells and the signaling mechanisms involved in nitric oxide production. So one of the first experiments that we did on this is to enrich the cells with cholesterol and look at the calcium response. And in this case, we're showing calcium response to ATP. And in the top left corner, the blue line is the normal response to shear stress, ATP, which is showing a calcium transient. The red line is in the response when we inhibit the store-operated channels with SKF. And we see that cholesterol actually inhibits that calcium influx even more severely than the channel blocker itself. And in the lower left corner, when we do that experiment where we stimulate in the absence of calcium and then we add calcium, we see that the effect of cholesterol is really most prominent on that secondary influx, that influx through the store-operated channel. So it seems that the cholesterol enrichment has a strong effect on calcium influx through those channels. In response to flow, the nitric oxide production is also impaired by enrichment with cholesterol. And then also under flow, we see that cholesterol inhibits the phosphorylation of enos. And so then looking at the phosphorylation of enos in response to ATP, we say, again, cholesterol appears to have a very strong inhibitory effect on phosphorylation of enos. And now going back to our model for the signaling pathway, I'll just remind you that we're interested in PKC, which we think stimulates the activation of the store-operated channels. And so we wanted to see, I'm leaving out a lot of data on the effects of cholesterol on different parts of this pathway to focus on the one that seems to have the most effect on it, and that is the store-operated channels. So if we treat the cells with a reagent that stimulates directly the store-operated channels, in the left side, you can see the calcium influx due to the stimulation with this reagent LPC. The calcium response doesn't look exactly like it does when we stimulate with ATP, but in terms of the kinetics. But nevertheless, when we enrich with cholesterol, there's a significant inhibition of the calcium influx due to those store-operated channels. And then when we look at enos phosphorylation, the direct stimulation of those store-operated channels does produce phosphorylation of enos. It's not as dramatic as ATP, but again, the calcium is not quite the same either. And nevertheless, the cholesterol appears to inhibit that phosphorylation. So just to try to wrap this up, some of the key findings from our work is that the transport phenomenon within our experimental system and also in vivo that we're modeling is strongly transport dependent. For the cell scale modeling, the caveolar-based enos signaling mechanisms are transport dependent and are incorporating more aspects of that as we go. Shear stress-induced NO production appears to depend strongly on ATP autocrine stimulation, and particularly this store-operated channel influx. And significantly for the pathological condition, it appears that cholesterol enrichment inhibits shear stress-induced NO production, and primarily through direct inhibition of these store-operated channels. So just a few things that we're pursuing now. We're going to build on that calcium model with the intracellular transport related to NO production by including not just direct activation of NO production by calcium-calmodulin interactions, but also mechanisms related to enos phosphorylation. We're going to look more closely at the role of PKC using some experimental techniques like SI knockdown and to complement what we've already done with pharmacological inhibitors. And we'll be looking at other pathways involved in enos phosphorylation as well. Importantly, we want to also drill down on this store-operated channel mechanism. And there are a couple of different channels that are implicated. One is ORI1 that interacts with a protein called STIM1 that's in the ER in response to depletion of calcium. TRPC channels, transient receptor potential canonical channels, are also implicated in SOC activity. And so we'll be looking at those using various experimental techniques and incorporating those into our model as well. And then finally, to integrate with the other models, we're going to try to incorporate the salient features of our cell scale models into the vessel scale and network scale models in order to reproduce some of that functionality. So now I will turn things over to Don Buerk, who does our animal experimental work.
Thanks, Ken. So I have had over 40 years of experience working with electrochemical microsensors, and wrong button. So most of my experience has been with oxygen electrodes, particularly the Whalen Nair type of recessed electrode. And the advantages to having a recess is that the electrochemical sensor is acting in a diffusion limited mode. So it's much, much less sensitive to convective transport disturbances. You can make smaller tips with etched platinum, but those electrodes tend to be very flow sensitive unless you put a very heavy-duty membrane. The advantage of the recess is you can get the membrane into the recess and further limit the diffusion. So most of my experience has been with oxygen electrodes, and someone once said, I think it was a grant review, said, well, all this guy does is measure oxygen, and I just decided to look into measuring other things. So I modified the Whalen type electrode to measure nitric oxide by changing the polarization voltage to a oxidation of nitric oxide, and furthermore put in materials in the membrane that goes into the recess to improve the selectivity. And this is an example of a calibration, 2-point calibration going between two different nitric oxide concentrations by bubbling nitric oxide in the solution. And if you can do a multi-point calibration by then injecting known amounts into a chamber and measuring the response. So the electrode is linear. It has excellent spatial and temporal resolution, so you can get very detailed measurements. So the in vivo preparation, among many that I've looked at, the rat mesentery is particularly simple. So there are flat surface. The surface vessels have pretty linear lengths. Of course, there are branching and there's interaction with other vessels, but it allows you to interrogate the surface with a microelectrode and be able to make measurements on the outside of the vessel wall. This system was developed when I was at the University of Pennsylvania. I had a laser Doppler probe, which was basically looking at the change in flow in the capillary region. I don't have access to that instrument. Now I'm going to use electromagnetic flow probes and put them on the upstream and downstream vessels. And I have been using a video imaging system that looks at the diameters. There's an edge tracking software. You can put a window on a vessel of interest and get about 10 samples per second of the dimensions of that vessel. And then you can do things to perturb the system, put in vasodilators in the bath surrounding the mesentery, or giving something to the animal, or changing the oxygen conditions in the superfusion solution. And one of the things I did is just simply try to map out what the distribution is along this network. And so this is an overlap of several images, and the circles show all the different points I investigated. The camera is a color camera, so you can tell the arterioles and venules quite easily. And what I did then is just place the electrode close to the vessel wall to get a measurement, and then pull the electrode back out into solution to get a 0 nitric oxide reference point. So the arterioles in the system, and again, you can tell these very easily by the color, are points A, B, D, F, G, and J. And the venules are shown. And then I just did one measurement in a region between vessels where there are lots of capillaries. And so this is an example of a measurement. In this case, I started out in solution and then approached the vessel. So this is the value that I found as I touched the surface of that vessel. And this is the diameter measurement. This is the smallest vessel in that map that I looked at 27 micron diameter. And when the electrode gets into the image, it interferes with that measurement. So when I do other types of measurements, I have to place the electrode a little bit downstream from where I'm doing the diameter measurements. And so this is the net result. And if you look at the different diameters, the nitric oxide levels, just in this table, it's not obvious that there's any relationship we can look at. The average for the arterials was 181.9 in this set of measurements. But if you start looking at the relationship between diameter and nitric oxide level, there appears to be this nonlinear relationship with very high values for the smaller vessels and smaller values for larger vessels. With this exception J, which I'll talk about in a little bit, this is actually where there's interaction between vessels. So again, if we look at the venules, it's actually a little bit higher in this set of measurements. And we have this same kind of relationship. It appears that the nitric oxide is higher with the smaller venules. And again, we have this is actually the highest value in this map. And that is in a region where there's interaction between the vessels. So K is a venule, J is a larger arterial, and they're very close to one another. And those 2 points were the highest nitric oxide levels in this particular animal. I've done this in the rat mesentery, but I've also done these measurements in a conscious hamster. So there's always concern when you have anesthesia that you're altering the conditions. And for control conditions, the values are quite high, above 500 nanomolar, and there's not much difference in these conditions, no significant difference between arterioles and venules. And the capillaries are a little bit lower. We transfuse these animals with different viscosity blood substitutes, and with a low viscosity solution where the shear stress would be altered, there was a decrease in nitric oxide, and actually there's hardly any difference between arterioles, venules, and capillaries in this animal. And then if we transfuse with a high viscosity solution, we've got a very large over doubling of the nitric oxide levels. And again, the capillaries have the lowest levels, and there's no significant difference between arterioles and venules. So we are going to extend these in vivo experiments, again using the rat mesentery. And we're going to do further characterization of nitric oxide and vessel diameter. And particularly, we want to look at the dynamic relationship between changes in nitric oxide and changes in vessel diameter, not just the steady state conditions. We're also going to look at the effects of ATP and the receptors, adrenergic receptors, and see how that changes that dynamic relationship. We're also going to be altering the oxygen supply to the vessels to preparation and examine the effects of hypoxia. And then finally, we're going to look at the role of nitrite and nitrite reductase. This is somewhat controversial. It's considered to be an alternative source of nitric oxide, although how it gets out of the red blood cell, if it's being produced by red blood cells, or whether it's coming more from the tissue, needs to be investigated. We're also going to move towards an animal model of this lipoprotein E deficient mouse, which spontaneously develops lesions, atherosclerotic lesions, and if you put the animal on a high-fat diet, it just produces much more lesions. So we're going to be looking at those regions. We'll have to take the vessels out and do ex vivo studies, and then we're going to look at the effects of vasoactive agents on nitric oxide production in those vessel segments. So that ends my part, and what I'd like to do before we open up for questions is just provide a little summary of what we're going to be doing. So this is innovative in the aspect that we are coupling a number of different mechanisms, and we're also, of course, doing this at the Multiscale scale level. We have novel instrumentation. We can produce measurements with cultured endothelial cells where we can control conditions to the cells. We also can do in vivo measurements, which are a little more difficult because you can't always produce as big a stimulus to the animal as you can in vitro. And we're going to be integrating this theoretical study with our experimental results. And we're also going to move towards a pathological model, and that is the cholesterol model for atherosclerosis. So that's a summary. So I'm ready for any questions you might have. Bring everyone up.
Thank you very much. Please, if everyone can unmute themselves and to unmute their videos, we'll start the discussion portion of this presentation. Are there any questions from the audience? So I guess I'll start with a question, a more generalized perspective question from the UO1 award. Is every UO1 award is to identify some problem that you're interested in addressing through the multiscale modeling community. So is there, what do you plan to do in terms of that aspect of the UO1 award?
Well, I am particularly interested in meeting other people who are actually developing techniques, mathematical techniques for scaling, for bridging these scales. So capturing the details of cellular signaling mechanisms in a model that was as big as all of the networks would be computationally prohibitive unless we have some clever way of doing that. So there are ways that people are developing to be able to capture some of the salient features of model behavior on a small scale and integrate into larger scales. That would be a big benefit to us.
Yeah, we are actually beginning to experience some challenges in performing very elaborate computations, and any help from the other participants would be very welcome. So
complicated computations in terms of mathematical methodologies or computational speed and possibly high performance computing?
In terms of the mathematical simulation and the linking of the different scales.
Grace?
Yes?
This is Herman Cavalier from the NIMH.
Okay, please feel free to show your webcam so we can see you.
I don't know if it, let me see. Is it, I don't see. Is it working?
Yeah, so you just place your cursor in the middle where the black box is.
The
toolbar.
Okay.
Great. And others, please do show your webcams so we can have a group participation.
But I don't see anything.
We can see you.
Okay.
Okay, go ahead with your question.
So my question was about the pharmaceutical, the statins. Did I miss where they change the cholesterol or the effects of cholesterol?
I'm not sure what you're asking. So are you talking about the cell in vitro model experiments?
These pharmaceuticals that are given to people to lower the cholesterol.
So we're not modeling that directly. So we're right now modeling theoretically and experimentally the effects of cholesterol on cell function. Presumably, if you lower blood cholesterol levels, you're going to lower the cholesterol content in the vascular cell membranes, and it should bring you back to a normal state. It's not clear what happens in the endothelium overlying a lesion. So if you have a lesion that's full of lipids, it may be enriched preferentially compared to other places in the vascular. The data that I showed was cells collected from blood vessels irrespective of the locations. We don't really know what the distribution is in terms of around a lesion or a non-lesion area. So the effect of statins certainly lowers blood cholesterol levels. Whether it affects more the endothelium of the lesions or just generally, I don't think anybody knows. I don't think anyone's looked.
And that's the reason for going to an animal model, because Ken's got some data with endothelial cells showing some impairment of nitric oxide production. And we expect to find the same in the animal model. And we're at the beginning stages of that. We eventually, I suppose, we could probably try to give statins to the animal and see if it helps. But that's not the focus of what we're doing.
Mm-hmm. Thank you.
Any other questions for our speakers? If not, we can continue the discussion online. I know your contact.
I have another question.
Okay, go ahead.
I'm curious about. I know where the oxygen comes, but where does it come from? The NO.
Where does the NO come from?
Yeah.
Primarily the endothelium. So in other tissues like brain, there's significant contribution from neural nitric oxide synthase, but in other tissues it's primarily the endothelium.
Uh-huh.
Or macrophages also make NO. So when they're fighting infection, that's how they kill bacteria, by secreting high concentrations of NO. So it's conceivable that in an atherosclerotic lesion that macrophages are contributing to that. But in normal vessels, it's the endothelium.
And there is a neuronal source for these blood vessels that I'm going to be looking at, and that's one of the studies. I can block that production. It's easy to block all production, but it's much harder to selectively pick out one or the other. So you can block out the iNOS with inhibitors, but you give enough inhibitor, then you start affecting the other nitric oxide.
You're talking about perivascular, no?
Right. Right. So there are places where there's going to be neuronal production. I can block that and look at what would mainly be from eNOS. And
We have also included iNOS in one of our models to show the effect.
Yeah, we've done the model, and the other thing is that nNOS, the neuronal one, is much more oxygen dependent than eNOS. So even if you have a small amount of nitric oxide production from a neuronal source, it could contribute quite a bit to an oxygen response.
Thank you.
Okay, great. Any other questions or comments before we sign off? If not, let's give our speakers a virtual hand. Thank you very much for your webinar, and we look forward to your participation. Thank you.